Hello, my name is Nicole Zeltser. I'm representing Cal Poly San Luis Obispo, and today I will be talking to you about my project, The Effect of Western Diet on Pediatric Brain Development using juvenile Iberian pigs as a model. The Western diet was introduced to the world after technological advances during the agricultural and industrial revolutions allowed easy access to foods high in saturated fat and refined simple sugars, such as fructose. As a result, children are exposed to such unhealthy diets early in life. A primary concern is the association of the Western diet with a host of conditions such as obesity and type 2 diabetes, which are associated amongst themselves as metabolic disorders. However, the effect of the Western diet on the brain, especially in children, is yet unknown. In humans, substantial brain development occurs before and after birth. While the number of neurons in our brains is set pretty much by birth, the brain still undergoes a significant period of development during the first couple years of life, during which it is vulnerable to environmental influences, including nutrition. This pattern of brain development is very similar to that of the domestic pig, but not so much to rodent models. The following pathway describing the connection between the Western diet and brain injury has been investigated in rodent models, but not in pigs or humans. So our subject, this mouse here, is exposed to a chronic diet high in fat and sugar. This causes insulin resistance and inflammation, which spreads to the brain. In the brain, inflammatory proteins called cytokines spread neuroinflammation. This is accompanied by the activation of astrocyte cells in a process known as astrogliosis. These factors, including brain insulin resistance, cause neurons to degenerate, which manifests itself as a cognitive impairment, such as memory loss. Our chosen animal model is the Iberian pig, which is particularly suited for this study due to a genetic mutation causing resistance to the hormone leptin, which prevents them from feeling satiated and predisposes them to metabolic syndrome. Our use of young piglets provides a pediatric model, and pigs in general are a better translational model than rodents due to similarities to human anatomy and physiology, including in brain development. Recently, the role of the gut microbiome in overall health has been receiving a lot of attention. Studies show that changes in the gut microbiota can influence insulin resistance, neuroinflammation, and cognitive behavior. Therefore, the ameliorative effects of probiotics have been the subject of much research. Thus, our two-part hypothesis. First, that feeding a high-fat and high-fructose diet for 10 weeks to juvenile Iberian pigs would result in brain insulin resistance and neuroinflammation, leading to brain injury manifested as neuronal loss and impaired memory. And second, probiotic supplementation will ameliorate the effects of the HFF diet. To test our hypothesis, we designed the following experiment. Piglets began receiving treatment diets at 13 days of age. They were fed every 6 hours by body weight for 10 weeks, after which they were humanely euthanized for frontal cortex sample collection. We had 4 treatment diets. The control diet without any probiotics added, or CON-N, a diet high in fructose and fat, again no probiotics, called HFFN, then the control diet with added probiotics, or CON-P, and the HFF diet with probiotics, or HFFP. And the sample sizes were 8, 6, 8, and 6 piglets respectively. We used Western blotting techniques to evaluate expression of proteins involved in the insulin signaling pathway, the ELISA assay to evaluate inflammatory cytokine levels, immunofluorescent staining to count numbers of neurons and astrocyte cells, the novel object recognition test to evaluate short-term memory, and liquid chromatography mass spectrometry to evaluate metabolites. For our statistical analysis, we used a two-way ANOVA mixed model with diet, probiotics, and their interaction as fixed effects, and the cage the pigs were assigned to as random effects. So the first thing we wanted to look at was inflammation in the brain. So we started with astrocyte cells. To do this, we took very thinly sliced pieces of brain tissue and stained them with a fluorescent stain targeted at the GFAP protein, which is specific to astrocyte cells. The stain shows up in red, and as you can see from these representative slides, you can actually visually see a significant increase in the activation of astrocytes in our HFF pigs. Next, we look at the presence of inflammatory cytokines. For this, we use the ELISA assay, which looks at small amounts of specific proteins. In our case, the cytokines, such as interleukin-1 alpha, represented in this graph here. And we did not find any significant differences between treatments. We were also very interested in brain insulin resistance. And for this, we used a Western blotting technique to look at expression and activation levels of proteins involved in the insulin signaling pathway, such as insulin receptor 1 here and protein kinase B here. And once again, we did not find any significant differences between treatments. However, having found some evidence of brain injury in our astrogliosis, we wanted to continue with this line of questioning. And the next thing we wanted to do was see if we could find a difference in the actual numbers of neurons in the brains. And for this, we went back to our staining technique. This time we targeted the NEUN protein, which is specific to neurons. And once again, the stain shows up in red. And we actually found a significant decrease in the number of neurons in HFF pigs. So now, having found evidence of physiological brain injury, we wanted to go back and see if we could find an associated behavioral change in our pigs. And for this, we used a novel object recognition test to evaluate short-term memory. During the test, the animal is presented with two identical objects for 10 minutes to investigate. Then an hour later, they are presented with two objects again. One object is identical to the first two objects, and one is a new or novel object. If the animal remembers already having investigated the old object, they are more likely to spend a longer time investigating the novel object. And that's what is presented in this graph here, which is showing the average amount of time the pigs spent investigating the novel object during weeks 7, 8, and 9 of the study during which the test was performed. And we see that in weeks 8 and 9, control pigs spent a significantly longer amount of time on average investigating the novel object. And this is an indication that the HFF diet may be causing some sort of memory impairment. Finally, we performed a metabolomics analysis to inform different potential pathways to neurodegeneration. We were looking for various metabolites that differed significantly between treatments. One significant result was an increase in simple sugars in HFF pigs, including sorbitol, which is a fructose to glucose metabolism intermediate. It was interesting to still see this sort of disruption in glucose metabolism despite not having found any signs of a change in the insulin pathway. This kind of accumulation in simple sugars could also potentially be toxic due to the oxidative stress that is put on the mitochondria. We also found a significant decrease in choline and betaine metabolites, both of which are involved in the production of the neurotransmitter acetylcholine, and also in the composition of the phospholipid neuronal cell membranes. And a disruption in either of these processes could also potentially be damaging. So to conclude, Iberian piglets fed a diet high in fat and fructose for 10 weeks developed signs of brain injury in the absence of insulin resistance, and no effect was detected from probiotic supplementation. Thank you very much for listening. I'd like to say a big thank you to my mentor, Dr. Magdalena Maj, who provided this opportunity for me, and a thank you to Cal Poly San Luis Obispo for providing all these wonderful facilities for us to use in pursuit of our research goals. I look forward to answering any questions you may have. Thank you for listening.